Tomorrow belongs to those who prepare for it today. A seed planted today takes root and grows into a tree that bears fruit in the future. Over 110 years ago, a seed was planted. Today, that seed, the idea of excellence, has grown into the University of Pretoria, South Africa's largest contact research-intensive university. 9 faculties and a business school are spread across 7 beautiful campuses, which are home to over 50,000 students, ready to make an impact in the world beyond university and join our global network of nearly 300,000 alumni. Future-focused, sustainably developed facilities and cutting-edge multi- and transdisciplinary research are underpinned by a desire to transform lives and have a positive impact on communities and the world. Excellence in teaching, learning, research innovation, arts and culture, and sports puts us firmly amongst the world's best universities. Knowledge is not just what is in books. It is the wisdom to apply it, to nourish and nurture the seed so that it takes root, grows tall, bears fruit, and branches out. UP plants that seed, that tiny bit of curiosity, creativity, critical thinking, hope, the desire to care, respect, help, and innovate against all odds, to grow, to leave your mark, to excel, to challenge the norm, to think, to rethink, to discover, to inquire, to lead, to have courage, to make a difference, and to persevere. This is the University of Pretoria. We make today and every day matter.
Good afternoon, ladies and gentlemen. My name is Tiaan de Jager, and I'm the Director of the University of Pretoria Institute for Sustainable Malaria Control. We are live from the University of Pretoria's Future Africa Institute and campus. Today we are commemorating SADC Malaria Day. This webinar, titled Integrated Vector Management and New Innovations to Limit Epigenetic Changes in Malaria Endemic Communities, was organized by the UP ISMC Student Forum. A special word of thanks to Danelle Oosthuizen and Mkubezi Mabuza. The University of Pretoria Institute for Sustainable Malaria Control is a fully integrated, transdisciplinary and interfaculty initiative. The aim of this institute is to coordinate and promote collaborative research on safer and sustainable malaria control and management strategies, and to generate new knowledge and support new activities pertaining to safe malaria control in Africa. The Institute hosts a DSI NRF SACHI Research Chair on Sustainable Malaria Control, a community of practice on malaria elimination, and is a South African Medical Research Council collaborating centre for malaria research. The Institute consists of 3 research clusters, including human health, parasite control, and vector control, with research occurring within and across clusters towards malaria elimination. The webinar is an initiative of the UP Institute for Sustainable Malaria Control Student Forum to commemorate SADC Malaria Week. The Student Forum aims to create a community amongst young and emerging researchers from different disciplines with a keen interest in malaria to bring together minds from transdisciplinary backgrounds that will be instrumental in pioneering solutions to end malaria. We have an exciting lineup with great speakers addressing a challenging topic. Our first presenter is Professor Liu Braak from Bangkok, Thailand. Professor Braak is a senior vector control specialist for Malaria Consortium Asia and the technical lead for the Asia-Pacific Malaria Elimination Network Vector Control Working Group. He completed his PhD in entomology at the University of KwaZulu-Natal. He was a research scientist in the Kruger National Park, covering general, medical, and veterinary entomology, but with particular focus on malaria. In 2012, he joined the University of Pretoria as research professor and is currently extraordinary professor associated with the UP Institute for Sustainable Malaria Control. His research interest is on vector biology, biting behavior of the main African malaria vectors, and integrated vector management with a special interest of innovative control measures. The title of his presentation is Malaria Vectors and Vectors Contact Reduction. Good evening, Professor Brock. We are looking forward to your presentation.
So, Tian, thank you for that, for those words of introduction, and great introduction by yourself. Very, very well articulated and good message there. To everyone, wherever you happen to find yourself on the planet, very good day to you. And thank you for the opportunity to share a few thoughts on this talk, as Tian pointed out, on malaria vectors and vector contact reduction. So I found that video insert quite interesting, where they made reference that University of Pretoria started about 110 years ago, if I remember correctly. So it's fascinating that it wasn't long before that that humanity first found out what the actual cause of malaria is. Malaria, of course, being a major scourge of humanity that has caused over time multiple tens of millions of deaths. So it was only in 1880 that Ivamectin in Algeria looked at the blood of a febrile patient and saw some of the stages of malaria parasites in there. And then it took another 17 years until Ronald Ross discovered in India that Anopheles mosquitoes are responsible for transmitting these parasites. And ever since that time, the control of malaria has by and large rested on vector control. So it demonstrates the importance that vectors play in the strategies to combat that disease. So just running through, since the time of discovery of what causes malaria and how it's transmitted. I'm not sure if you can see my cursor, but soon after the discovery that Anopheles mosquitoes are responsible for the transmission of malaria, the first work done, it was in Italy by this Angelo Celli against vectors, by means of house screening and burning of powders, probably pyrethrum, which demonstrated a dramatic reduction in incidence of malaria. And it showed the efficiency of targeting the vectors for malaria control. I find this a particularly fascinating little story, that the Panama Canal in Central America, the French got the opportunity to build the Panama Canal, and they started way back in 1881 again. But because of problems, management problems, and also the exceptionally high levels of mortality, people dying from mosquito-borne yellow fever and malaria, they had to ditch the project, literally ditch the project. The project was led by Gustave Eiffel, the same guy who built the Eiffel Tower in Paris. And his reputation took a nosedive because of the failed attempt at building the canal. However, when it was learned what was actually the cause of malaria, the United States took over the project in 1904, And they finished the canal in 1914. And it was made possible by the dramatic reduction of malaria and yellow fever by, again, vector-based interventions such as house screening, larviciding, combating the larvae in breeding places by spraying oil on the water, Paris green, fumigation with the pyrethrum, draining breeding sites, removing the forest where the mosquitoes were finding refuge, and so on. And then there were subsequent very successful malaria campaigns focusing on vector control. Oh, I can't see my cursor. There we go. Such as in Malaya, now called Malaysia, 1901 to 1920, again by means of vector-targeted interventions, draining ditches, etc. Then we move on to the Second World War. And early in the Second World War, DDT made its appearance, and it was a massive relief that it was very effective in controlling lice, lice-borne typhus, which was a big scourge and causing a lot of deaths amongst the troops and so on. So they then transferred the use of DDT then to agriculture and also for malaria control. Also in the Second World War was the rediscovery of the efficacy of chloroquine, then called Resokine, in the early 1940s against the malaria parasites. So with this dual combination of, call it silver bullets if you like, of DDT against the adult mosquitoes and chloroquine against the parasites, the world then thought, okay, now we have the tools to eradicate malaria off the face of the planet. So WHO launched a global malaria eradication program from 1955 onwards, mass application. Here at the bottom right, you can see aerial application of DDT and mass application of DDT and other insecticides, and then also large-scale application of chloroquine, dual combination of techniques to fight malaria. But inevitably, resistance set in, and woefully, by 1969, WHO had to abandon the Global Malaria Eradication Program, and they switched to calling it a malaria control approach thereafter, but still holding out hope and saying that, don't worry, we'll eventually eradicate malaria, we'll get there. Although this global malaria eradication program was abandoned, those earlier years did result in remarkable successes, such as eliminating malaria from the southern parts of North America, the United States, southern parts of Europe, Egypt, and interestingly, Anopheles gambiae, which is a major vector in Africa, had somehow found its way into Brazil, South America. And again, that global malaria eradication control program or eradication program was successful in eliminating gambiae from South America. So it wasn't a wasted effort, much was achieved. Then we move on until the end of the 20th century, by which stage larviciding and house screening and so on, and by and large been abandoned in favor of two strategies. Insecticide-treated bed nets and indoor residual spraying. Put everything into those two methods and mass distribution of hundreds of millions of bed nets, supported to some extent by indoor residual spraying. Focus was largely on provision of bed nets. And together, these two techniques, which target mosquitoes coming indoors, very narrow target, you're targeting those mosquitoes that come indoors to bite people at night inside the household. Together, those two were highly successful in reducing malaria numbers globally, at least in the tropical belt or malaria endemic countries, from 2000 up till around about 2015, and then reached a plateau. And we are still in that plateau. In fact, malaria is in many countries is actually increasing. So this is very, very disturbing, obviously, because, you know, we're pouring all our available tools at malaria, and we're not gaining much from all that cost and time and effort and so on. So this is the trajectory, the green line, that should have been followed, but then we hit this plateau over here. So unless we come up with innovative thinking, new strategies, and particularly new tools, the red line shows where we are going to head then, back up by 2030, back up to about 2000 levels again. What is more likely to happen is that we'll follow an intermediate path. We'll come up with some new bright ideas to overcome some of the problems, and we'll likely follow this dotted line path, a more gradual reduction in malaria cases, hopefully reaching global eradication of malaria by about 2050. Those are the projections at the moment. So what went wrong? By and large, you can attribute it to insecticide resistance by the mosquitoes to the particular chemicals used in insecticide-treated bed nets and indoor residual spraying. In the earlier years, the focus was largely, not exclusively, but largely on synthetic pyrethroids. And mosquitoes tend to develop a resistance fairly rapidly against synthetic pyrethroids. And this is a problem because you can't use some of the other compounds because humans are in close contact with the bed nets, for example. So you have to be careful what chemicals you use in those bed nets. So together with this insecticide resistance, the residual malaria that remained after you've, hammered the mosquitoes with these bed nets and indoor residual spraying, much of the residual malaria was caused by outdoor biting mosquitoes, where we don't really have any tools. We poured all our effort into indoor techniques, and now we sit with the problem of outdoor biting, and we don't have tools to address that, not really effective tools. So, but there are also, there's a new generation of chemicals and long-lasting insecticide-treated bed nets coming to the fore, becoming available, that use completely different classes of insecticides and also use synergists, piperonal butoxide, etc., that renew the efficacy of those bed nets. But you still have those outdoor biting mosquitoes that cause residual malaria. So let's have a quick look at the malaria vectors at play in Africa. The prime vectors, these are the two best malaria vectors on the entire planet, Anopheles gambiae, sensu stricto, and Anopheles funestus. And both of these species are strongly attracted to humans and strongly enter households. And that is what makes the bed nets and indoor residual spraying so highly effective. So these two remain the very best vectors operating throughout much of Africa, but there are others. Anopheles arabiensis is worth mention. It's a major vector, especially in the southern parts of Africa, but also many other parts of Africa, Ethiopia, etc., etc. Mainly in the inland savanna areas, it's a major vector, and this particular species bites both indoors and outdoors, and it looks for blood both at animals and in humans. And of course, this makes it much more difficult to control. You can't narrowly target them indoors. So this is a relatively small number of species compared to, for example, the Asia-Pacific region, where there are many different species complexes within which you find a number of different malaria vectors. And with most species, what's happened with my cursor now? With most species feeding as readily outdoors as indoors and as readily at animals, such as cattle and water buffalo, as at people. If you look at the little map here at bottom right, you'll see in South America, if you look at the colors there, each color representing a different vector species, relatively few species responsible. In Africa, few more, but in Asia, there's a whole cluster of different species of malaria vectors at play. So we look at Southern Africa, since we're talking about University of Pretoria and its role in combating malaria. The main vectors at play here along the, for example, Anopheles funestus along the coastal belt of Mozambique. It also occurs in Zimbabwe and a few other places. But by and large, this is its main area of distribution. In the inland areas, it's largely Anopheles arabiensis. That's the assumption. So, but it's interesting, and it's probably true that in much of southern Africa, Anopheles arabiensis is the major vector, but it doesn't hold homogeneously across the inland savanna regions, malaria endemic savanna regions. For example, the University of Pretoria led a survey in this northeastern corner of South Africa, and in particular region of that, the Limpopo River Valley. We found Anopheles arabiensis is in fact extremely rare. And it's not through lack of efficiency of the trapping methods we use, because we use the same methods elsewhere and with great success for Anopheles arabiensis. Anopheles arabiensis is very rare over here, whereas certain members of the Anopheles funestus complex, Anopheles funestus group, are very abundant in that region. So, and the wonderful work, the excellent work done by the group at the National Institute for Communicable Diseases in Johannesburg, they found that certain members of the funestus group, such as Anopheles paurensis, Anopheles van Edenai in KwaZulu-Natal and Mpumalanga, do have gametocytes, do have sporozoites inside them, and are almost certainly playing a very significant role, a previously unsuspected role, in the transmission of malaria. And this is almost certainly what is happening in Limpopo province. That transmission is not primarily due to Anopheles arabiensis, but to these outdoor biting, animal feeding, occasionally human feeding members of the Anopheles funestus group. So let's digress a bit and look at other, what I find very interesting aspects of malaria vector feeding and biting behavior. It turns out that across the planet, as indicated by multiple different studies, the malaria vectors, irrespective of which species they are, all have a very strong preference for feeding on ankles and feet. So in a study that we conducted in Uganda and in South Africa, we found that for Anopheles gambiae, Anopheles funestus, Anopheles arabiensis, all three species, about 95%, well over 90% of them, bite below, primarily at ankles and feet. Here's an example, and I think my good friend Mtoko is going to use the same slide in his talk coming up next. You can see this very strong convergence of biting at ankles and feet at people sitting or standing outdoors. And this was picked up intuitively by teams of people that are sent out to go and do vector surveillance, collect vector mosquitoes to monitor species composition and abundance of the malaria vectors out there. You look at these people busy with their activities and you'll see they focus on ankles and feet because they've learned intuitively this is where those mosquitoes are biting. So the interesting thing we found in our studies in Uganda and South Africa is that if you cover your ankles and feet and you prevent those mosquitoes from accessing ankles and feet, those mosquitoes don't move up to the exposed areas and go and bite there, as would be the obvious thing to do. They rather fly off, they fly away to find somebody that does have exposed ankles and feet. Very interesting and very relevant when you're thinking of exploiting this very strong behavioral factor for vector control. So I said, at people sitting or standing outdoors at night. However, if you take a volunteer and you put that person flat on the ground, the situation changes dramatically. Mosquitoes now, the vector mosquitoes, will bite you anywhere on the body, which tells you that it's not an inherent preference for ankles and feet, it's a height above ground issue. The malaria vectors, whether in Papua New Guinea, Thailand, North Africa, Southern Africa, wherever, those mosquito species prefer flying and feeding close to ground level. So if you look at how rural people dress in most of the rural areas of the tropical belt, typically, especially women and children, they walk around night and day with bare ankles and feet. And these are exactly the areas that those malaria vector species are targeting. So, I mean, people buy these mosquito repellents and they happily go and apply it all over their arms and neck and face and so on. That's useless. This is where the malaria vectors are focusing their attention. So there were some interesting correlated studies relating to this. So, and a study that I did in Kruger National Park in early 90s already, putting tables on top of each other and getting volunteers to sit at different heights. But here's a publication by Charlwood in 2003 saying that the ancient Egyptians were right. People in houses raised on stilts suffer fewer mosquito attacks than people living on the ground. So for Anopheles gambiae, he showed that at a house on the ground, you were getting about 68 bites per collection, whereas in a house 2 meters off the ground, you were getting 3.7 bites per collection period. Similar in Kenya, where a great difference in the number of mosquitoes, 5 per collection in the houses raised off the ground versus 30 when on the ground. Here's another one, also in Anopheles gambiae, control house at ground level. If you raise yourself 1 meter above ground, 41% bites, 2 meters off the ground, 68% less bites, 3 meters off the ground, 84% less bites. So I need to speed up because I realize I'm approaching my time. So what happens indoors? Those mosquitoes are almost certainly, I'm not aware of any study that's looked at this, but the mosquitoes are almost certainly flying close to the ground. They hit an obstruction. They then go boing, boing, boing up to get over the obstacle. They hit a gap in the mosquito net and they enter. Or they fly around outside, they hit the wall of the house, and then they fly up. It's a trait associated with the malaria vectors recorded by Snow in 1987 already. They bounce up trying to get over the obstacle. They get to the eaves, they smell the odor from inside, and they come in through the eaves. But their natural preference would be close to the ground. So rural people in communities across the world have picked up on this, and you find in many communities they've learned, build a little platform, raise yourself off the ground, and you reduce the number of bites that you're being subjected to by mosquitoes. This is in the Gambia in a publication in 1989 already. So we mentioned that we are stuck with a problem of we've reached a plateau in terms of the use of bed nets in IRS. We've bludgeoned indoor frequenting mosquitoes as hard as we can to the limit of what we're able to reach. Now there's residual malaria, largely due to outdoor transmission, but we don't have the tools. Fortunately, people have started paying more and more attention to this, recognizing the shortfalls, and there are a number of products in the pipeline that have already, in some cases, been trialed, a lot of them in Africa, and they include things like topical and space repellents. My good friend Mtoko is going to talk about some of that in his presentation a little later. Toxic sugar-baited stations. The female mosquitoes still need access to sugar meals like the males. So they don't only take blood, they also need a sugar meal, such as that nectared flowers. And when they're attracted to these sugar-baited stations, they pick up the insecticide in the sugar bait and are killed. Saturation trapping of mosquitoes. Then you get to several high-end, very specialized techniques that are more likely going to find very specific focal applications, such as sterile insect techniques, mass release of sterile males to outcompete the wild population of fertile males, Wolbachia, which are naturally occurring bacteria, and they reduce the ability of pathogens to reproduce. And then you get these genetically modified mosquitoes where you introduce genes into the population of mosquitoes that reduces the transmission of parasites. So I mentioned the space repellents. Here's an example of work that's being done in Tanzania, impregnating strips of material, Hessian cloth with flumethrin, and then you wrap it around the area that you're sitting in outdoors, and the odor given off by the flumethrin, transfluthrin rather, repels the mosquitoes. And they've gone a step further and taken this, they've made strips of transfluthrin impregnated material, attached it to the underside of chairs, it also has a repellent effect, or pasting strips of transfluthrin impregnated Hessian onto sandals. So all kinds of applications for these transfluthrin impregnated materials. And it's been done in Vietnam, so it's catching on. The problem is it's also a pyrethroid, and mosquitoes develop resistance fairly easily against pyrethroids, so we need to see what happens there. A method that I haven't mentioned that I'm very excited about is the use of Ivamectin. So Ivamectin is a product, let me go to the next slide. It's been used for decades already against control, or for the control of onchocerciasis and lymphatic filariasis. Lymphatic filariasis is this elephantiasis, all mosquito-borne. And so people were treated with Ivamectin to kill the little worms that cause this onchocerciasis in the eye. There's a worm there in the eye, and the worms causing nematodes, causing filariasis. And then, of course, Ivamectin is also a very bread and butter kind of product, widely used across the world for worm control in cattle, very routine product. And people started noticing that, mosquitoes feeding on people and cattle recently treated with Ivamectin die. So long story short, there are now new formulations of Ivamectin, slow release, long-lasting formulations where if you apply it, inject it into a cow, then mosquitoes feeding on that treated cow for as long as a month up to a month, you will have 100% of mosquitoes feeding on that cow dying. And then, of course, a gradual diminishing after a month as the dose in the blood diminishes. So this is a method that holds great promise because, at least in Asia, in rural areas, every third, fourth house, people have 2 or 3 cattle that are in very close association with the household. So you can imagine treating this cow with Ivamectin, and for the next month, every single mosquito that feeds on that cow will die. And given that the vectors in Asia readily feed at cattle, this is a wonderful way to contribute to malaria elimination objectives. Cattle are also a very frequent feature of the African landscape, not quite underneath your house, different cultural habits in Africa, but still cattle are very frequently associated with villages and so on, and well worth trying out as well, where you have cattle maybe dispersed throughout the village, treat them with ivermectin, and you get a huge impact on mosquito populations that way, especially species like Anopheles arabiensis, and also those members of the Funestus group that feed on cattle. So I'm almost done. We should be diversifying our interventions during the elimination phase to target and mop up as many contributors to residual malaria as possible. I mentioned early on that since the discovery of the causes of malaria, malaria control has largely been achieved through vector control. When we reach the malaria elimination phase, pre-elimination, we need to take a more integrated and holistic approach that combines vector control with parasite control, and very definitely a much better effort at community engagement for community participation, so that communities who are the ultimate beneficiaries of malaria control programs, so that they actively participate and support malaria control. So thank you, ladies and gentlemen. Thank you for your patience.
Thank you, Leo, for a really thought-provoking presentation and for highlighting the importance of innovation. This should help us to get on the road to malaria elimination eventually. To our viewers, please remember to pose your questions in the box below the video, and also follow us on the social media platforms. Ladies and gentlemen, our second presenter is Professor Sarah Kimmins from Montreal in Canada. Professor Kimmins is an associate professor in reproductive biology in the Department of Animal Science in the Faculty of Agriculture and Environmental Science at McGill University. She received a PhD from Dalhousie University and completed her postdoctoral training at the Institute of Genetics and Molecular and Cellular Biology in Strasbourg, France. She is an associate member of the Department of Pharmacology and Therapeutics, Faculty of Medicine at McGill University. She holds a Tier 2 Canada Research Chair in Epigenetics, Reproduction and Development. Her research team is focused on understanding the molecular mechanisms underpinning epigenetic inheritance. Particular interest is given to paternal health and how environmental exposures alter fertility, clinical outcomes, and the sperm epigenome, development and health of offspring. Her presentation title is Endocrine Disruptors and Health Effects in Africa due to insecticides used for malaria vector control. A very good morning to you, Sarah, and welcome.
Good morning to you. Thank you very much for the invitation to speak today. I'm going to tell you a little bit about how these exposures in the environment, including insecticides, can impact things like fertility and perhaps even the health of the next generation through changes in the sperm epigenome. I just want to make sure you can see my screen. Okay, great. And are these slides that I'm sharing? Can someone just confirm that?
We can see.
Yeah, and they're my slides, right? From my computer?
We can see.
Okay, great. So I'm going to introduce you to this idea of non-genetic inheritance. So as I mentioned, we're really focused in my research group on talking about the paternal origins of adult disease. So that's basically how can exposures to the dad influence the health of the offspring. So this idea of non-genetic inheritance was really first presented by two researchers in Sweden, Gunther Kati and Marcus Pembry. And they engaged in two epidemiological studies that are really noteworthy. And I'm going to tell you about one. So if you look over on this map, there's a very remote region in Sweden known as Överkalix. And the researchers examined food availability in that region. And something specific to the area was they kept incredible records of things like the amount of food, the amount of crops grown, and really good birth and death and health records. So they went through that data and they studied a period where there was good food availability. And then they looked at the offspring and even the grandchildren. And what they found is when they compared 20 years of good food availability in those descendants to 20 years or 21 years of poor food availability, was that if you had a child during what we call the slow growth period of one of those fathers who were exposed to good food or poor food availability, they differed in their health. So before puberty, too much food in the grandfather suggested that there was a risk of diabetes and heart disease and reduced lifespan in his descendants. So that suggests that just when you're starting to develop sperm, if it's exposed to different variations in food availability, this can affect descendant health. But how this occurs at this time was really not known. We believe it's through the epigenome, and so did the study authors. So welcome to the epigenome, which is the study of why your DNA is not your destiny. So there's lots of excitement when we study the epigenome because you can alter it. It can respond to changes in your overall health and be improved. What's not so clear is if you alter your health, can you improve the health of your future children? So to look more at this definition of epigenetic inheritance and what we study, it refers to marks that are passed through the gametes, being the sperm and the egg, to alter gene expression in the embryo and in turn, the long-term health of the offspring. The epigenome includes the methylation of DNA and alterations of histone proteins, including methylation on lysine. So if you look here, this is a schematic that's showing you DNA that's associated with the nucleosome. And the nucleosome is made of 147 base pairs of DNA that are wrapped around a histone octamer. And these little lines projecting show you the tails of the histones. And we study the modifications on those tails and how they impact gene expression. Another mark that's important for the epigenome that I'm not going to talk about today is DNA methylation. So if you look closely here at this schematic of a nucleosome, you see the DNA in blue, and then you can see the histone tails here that are protruding from that nucleosome. We're particularly interested in histone H3 and its modifications on the tail, including methylation. What we found in our previous animal model studies is that if you alter the levels of histone H3 methylation in sperm, it's associated with altered offspring development and altered offspring health through things like metabolic disorders. So we have done various studies where we've altered the quality of the diet in mouse models. So for example, diets that are folate deficient or high-fat diets. And we associate these diets with changes to the sperm epigenome in the sires, which then lead to intergenerational changes in the health of the offspring. And we can observe some alarming effects when you change the sperm epigenome, such as an increase in birth defects, pregnancy loss, and then as the mice age, an increase in metabolic disorders if the father was on a high-fat diet. So basically this field that we're working in, we've termed it Pohad, which stands for Paternal Origins of Health and Disease. And we ask if paternal exposures can predispose future generations to birth defects and complex disease, for example. We've done a lot of this work in animal models, but more recently we've begun to work in men. And I'm going to show some of that studies we've been doing in collaboration with Professor De Jager in South Africa. But first, I want to tell you about our overall hypothesis. So if you look at the schematic here, we believe that exposures to the father can alter his fertility and potentially the sperm epigenome. When you have changes in the sperm epigenome, at fertilization and then through embryo development, this can lead to changes in gene expression in the embryo. These gene expression changes through development can then predispose the offspring for things like complex diseases and altered phenotypes. So we need to start thinking about sperm as being much more than just a vehicle for the paternal genome. Sperm does a lot more than transmit just the DNA. So the epigenome marks in sperm that take the form of DNA methylation or histone modifications, these are associated with the control of gene expression during sperm development. So if you change those marks in the sperm as it develops, you can impact a man's fertility. More recently, we've determined that not only can you affect a man's fertility through exposures, you can affect the epigenomic map that is transmitted that guides embryonic gene expression. So in some studies, we have shown that the histone modifications that are changed in the sperm, those modifications are retained in the embryo. And that is how they can influence gene expression through development. So this sperm can transmit what we call an epigenetic memory of environmental exposures. But the mechanisms of how it does this are still poorly understood, and there's been very few studies in men. This is Dr. Michael Skinner. He's a professor in the States at Washington State University. And he was one of the first people in the field of epigenetic inheritance to show that there can be transgenerational effects, similar to what I mentioned with the Gunter Kadi study and the food availability at the beginning of my talk. So basically, his work was transformational for the field because it showed that if you expose a male mouse to an endocrine disruptor, beginning in utero, the impact on the descendants includes not only that mouse's first generation offspring, but also the second generation offspring, which means it's transgenerational. So to describe that study in a little more depth, he used a rat model to study the reproductive effects of endocrine disruptors. Endocrine disruptors are chemicals or molecules that interfere with hormone signaling. So they would interfere, for example, with androgen receptor signaling and estrogen receptor signaling. You can find these everywhere in the environment. So they're used as industrial solvents and lubricants, and their byproducts include things that are damaging to, for example, metabolism. Some things like BPA exposure have been associated with increases in BMI. So some things that we know are endocrine disruptors are bisphenol A, plasticizers like phthalates, PCBs, and polybrominated biphenyls. So in this study, he exposed to two endocrine disruptors, the pesticide methoxychlor and the fungicide vinclozolin. He injected pregnant rats with these chemicals, either one or the other, during the second week of gestation. And this coincides with a period of development of the gonads, so development of the testes. And it's also a window where you're establishing part of the sperm epigenome that will be inherited. So what he found is that the F1 generation male offspring exposed in utero had abnormal testes with very few sperm. What was interesting was the generation that was not directly exposed, being the F2 generation, so the grandchildren, or the F2, being not directly exposed, had the same phenotype. So this indicated that exposures, for example, to a grandfather, if we're thinking about men, could impact the grandchildren. So it was the first study that showed transgenerational exposures can occur. So we've been looking at, now this is years later, and we've gotten much better at molecular biology and sequencing of the genome. So we've really been able to drill down, getting closer to understanding the epigenetic mechanisms than that first study of Mike Skinner. But he really opened the field to this idea that, hello, can you still see me?
Yes, we can.
Okay, I just lost the image. So this has really opened the idea to further study, not just in animal models, but in men, this improvement in techniques, and looking at all sorts of different environmental exposures and what are the impacts on the defendants. So animal models that exist today that have been very well performed indicate that indeed environmental exposures to things like BPA, phthalates, and other endocrine disruptors, as well as lifestyle factors like diet, can impact the sperm epigenome. Whether these have transgenerational effects is still questionable, and more studies need to be done, for sure in men. So we've looked at things like diet, and we show that if you expose to high-fat diets or folate deficiency, this can lead to intergenerational effects, not transgenerational. Other colleagues have looked at endocrine disrupting chemicals, things like a mix of what you would find in the Canadian Arctic, which includes PCBs, among other chemicals. And this is work of Janice Bailey. And she's shown that you can have transgenerational effects of exposure to a mix of endocrine disruptors. So this suggests that this is important work that should really be explored in men. So what do we know about exposure to insecticides that act as endocrine disrupting chemicals? We know that in certain regions, like in the Canadian North, in our Arctic, as well as in Greenland and in South Africa, that exposure to these chemicals can be associated with increased health risks in men and children. So exposures are associated with reductions in male fertility because of the endocrine action. In some of these populations, like in Canada's North, there's a higher incidence of miscarriage, stillbirths, poor fetal growth, birth defects, and urogenital malformations have been observed in South African populations. In Canada's North, exposure of parents has been associated with neurodevelopmental delays in children. So we're quite interested in the effect on birth and the health of the children in our lab, because this is something we see in Canada's North, but also something we see in our mouse models. We often see birth defects in our exposure models. So in the world, birth defects occur in about 2% of births and are a leading cause of infant death. In regions where there are higher rates of insecticide being sprayed or higher rates of insecticide exposure through the way these things move within the earth, such as in Canada's North, there are higher rates of birth defects. And so 20% of birth defects are caused by environmental agents, and we're very interested in whether some of these environmental agents can work through the fodder to increase birth defects. So we've been working with colleagues and a team led by Professor De Jager to study what happens in the Limpopo province of South Africa, where we know there are exposures to various insecticides, to a mix of insecticides. So we looked at men, 197 that were recruited from 2016 to 2017 in villages that use indoor residual spraying compared to villages that are not sprayed. What we found was really intriguing, and to my knowledge, it's the first time that an exposure has been associated on the sperm epigenome with changes in histone methylation, but that it occurs in a dose response fashion. So if you look here, shown here in the yellow, orange, and red, we measured in the sperm by in-depth epigenomic sequencing of histone H3, and we compared the enrichment levels in the non-sprayed villages and sperm from the non-sprayed villages. And what we found is that the higher the body burden of insecticides, the greater the response to the sperm epigenome. So there were regions in the sperm that increased in our target histone H3 trimethylation, and regions that also decreased in histone H3. It was the regions, both these regions, that were dose responsive. So when we did an analysis of what kind of genes and processes and functions they're involved with were altered in the sperm, we found that there were processes involved with reproduction, fertility, embryo development, learning and memory, and things like social behavior and neurodevelopment and vocal learning, for example. So this suggests that perhaps changes induced in the sperm epigenome, if they are retained in the embryo, which is the big question here. We don't know if that happens in men like it does in our animal models. Could this in turn have an effect on the population health? So these are things that we still need to study, but this is really suggestive that endocrine disruptors may not only impact a man's fertility, which is known, but may also impact the health of his future generations. So to conclude, we have shown for the first time that there's an association of insecticide exposure in men with an altered sperm epigenome at the level of histone methylation, and that there's potential here to affect offspring health. Our study findings indicate that there is a pressing need to find alternative insecticides that do not have epigenetic altering effects, and that before such things are used in the environment, there needs to be adequate toxicology studies to ensure that these alternatives are safe and non-epigenetic impacting. I'd like to thank the members of my research lab who've been involved in these studies, the animal studies, as well as the studies in men. Our collaborators here in Canada, Janice Bailey at the University of Laval and Amanda McFarlane at Health Canada. Our funding also, and then of course we're grateful for the participation of the men in the Limpopo community and the community workers who are involved in those studies, and the team that's led by Professor De Jager at the University of Pretoria. Thank you very much.
Thank you, Sarah. Thank you for not only getting up early this morning, but also sharing this different perspective of malaria control with our audience. This is well aligned with the aim and the vision of the University of Pretoria Institute for Sustainable Malaria Control in terms of safe and sustainable malaria control methods towards elimination. To the audience, remember, you're welcome to put your questions in the box below the video. Our final presenter is Dr. Mtoku Sebanda, right here in the Future Africa studio in Pretoria in South Africa. Dr. Sebanda is the Chief Executive Officer of African Applied Chemical and a University of Pretoria alumni. He holds a PhD in chemical engineering, specializing in polymers and chemical product design from the University of Pretoria, where he also completed his postdoctoral training. In 2016, under the mentorship of Professor Walter Fokker, the research group made a significant scientific breakthrough in the slow-release technology of liquids from fabric yarn. The group patented this technology, and this led to the founding of a startup company called African Applied Chemical, which focuses on applying chemical engineering technologies to solve problems in Africa, also related to malaria, of course. This presentation, titled Development of Innovative Vector Control Tools at the UP Institute for Sustainable Malaria Control. Thank you, Dr. Sabanda.
Thank you, Tian, for that introduction, and thanks to the audience for joining this webinar. This is going to be the overview of my presentation. I'm just going to talk a little bit about the mosquito and the disease it transmits. I think Leo Brack has also touched at length on that, the problem of outdoor malaria transmission, just to add on to what Professor Brack has presented. And we're going to talk also about vector control methods that are currently being used, and a summary of the techniques, or innovative vector control techniques that we developed at the UP Institute for Sustainable Malaria Control, whilst also working at the Institute of Applied Materials, which was collaborating with UP ISMC. And I'm just going to pick one of the flagship technologies that we developed and is currently undergoing commercialization. Okay, so this is a very beautiful image of a mosquito taken using a scanning electron microscope. It's a very powerful microscope, and it managed to capture a high definition image of this. Close up is a very beautiful insect, but responsible for a devastating disease in Africa. Okay, let's just speak in summary of the diseases that mosquitoes transmit. We've got dengue fever, chikungunya virus, yellow fever, and the Zika virus. But the major disease that mosquitoes transmit is, of course, malaria. Okay, 90% of the deaths, worldwide deaths due to malaria, are in sub-Saharan Africa. And another chilling statistic is that about 74% of those deaths that occur in sub-Saharan Africa are in children under the age of 5. So we have to work very hard to find a solution in suppressing malaria and eventually eliminating it, particularly from Africa. Okay, so Leo has talked at length about malaria control. Basically, the techniques are divided into two: parasite control and vector control. Our group was comprised of chemical engineers and chemists, and we focused mostly on vector control, or on the mosquito that transmits the parasite from one person to the next. So Leo has spoken at length about the various techniques used in vector control. What I wanted to add on to was indoor residual spraying is used mostly in South Africa. It's used with DDT, and long-lasting insecticide impregnated nets are used in the rest of Africa. Other lesser techniques is larviciding that Leo has also spoken about, and you can also use other sustainable techniques such as biological control, such as introducing probably fish that feed on the larvae, or you can just drain the standing pools that are used by mosquitoes to breed. Okay, so I'm just going to go through the various innovative products that we produced at since we started working on malaria research at the Institute of Applied Materials under Professor Walter Fouke. Basically, I joined the group in 2009, and Walter had obtained a Bill and Melinda Gates Foundation grant of about $100,000 to develop an alternative formulation for use in indoor residual spraying. We wanted to substitute the use of DDT, because DDT is used in South Africa to spray in people's houses. And there's a lot of controversy surrounding DDT, and as the last speaker has shown us, indeed DDT is responsible for adverse health effects on people living there in the Limpopo area where it's being deployed. And we came up with a formulation that could be sprayed, and we show that it's as good as DDT, and it culminated into a paper that was published in 2011. And then Walter was already working on a polypropylene-based long-life insecticide-treated net, and he was using polypropylene simply because Sasol produces quite a huge amount of polypropylene, and you can get access to it very cheaply here. And I managed to produce such a product. And then we moved on from that polypropylene-based net into wall linings, or monofilament mesh linings that were also treated with insecticides. The idea behind these wall linings was, instead of spraying the walls with insecticide as used in IRS, we can line the walls with a treated lining, treated with a sufficient quantity of the insecticide, and treat the upper parts of the wall instead of treating or lining the wall. So we did a huge field study in Limpopo, in the Vembe district, and there is a paper by Tanishka Kruger that was published in 2015. And then we moved away from researching on devices that use insecticides to devices that use insect repellent, simply because from the studies that Leo Brack had published showed that a significant amount of malaria transmission is occurring outdoors rather than indoors. Mosquitoes had changed their behavior to prefer to feed in the early evening and avoid going indoors and bite people before they go into their houses. And also the mosquitoes that tend to prefer to feed outdoors were selected by these techniques that focus on mosquitoes that feed indoors. Mosquito nets and IRS focus on mosquitoes that feed indoors, but the mosquitoes that feed outdoors were really were not focused on. And the theory is the reason why there's a plateau in the reduction of malaria cases is because the mosquitoes that feed and rest outdoors are now playing a prominent role. And Walter, with the student Homa Izadi, produced a promising mosquito repellent formulation that was supposed to be stable compared to the current commercial formulations that are available. Because if you apply an insect repellent on your skin, it lasts for about an hour or two before it evaporates. But the formulation that they produce showed that it can last for more than 6 hours after being applied on the skin. But what I want to focus on in this presentation is a breakthrough technology that we developed with some colleagues from Germany, whereby we could manufacture long-lasting insect repellent textiles. And these textiles could be used to manufacture articles that can then be used to protect selected parts of the body where mosquitoes that feed outdoors and can transmit malaria usually bite. Okay, so Leo has shown that mosquitoes that transmit malaria, they tend to bite on the lower limb when they are outdoors. They prefer to fly close to the ground, and in this case, if you are standing, they go for your ankles and feet. So it might be, if you have a device that is insect repellent and is worn around the the ankles and feet, you will reduce infective mosquito bites by up to 93%. Remember, transmission of malaria is a game of probability. If you reduce the number of mosquito bites, you reduce the probability of transmitting malaria, and therefore reducing the transmissions. You've seen this image from Leo's presentation. These mosquitoes, they just swarm and sit on your ankle. On the right-hand side is my foot. We're doing lab studies. These are lab-reared mosquitoes. There are about 300 mosquitoes in a cage. I'll just show you the cage as we progress with the presentation. And when you put your foot in that cage, the female mosquitoes just swarm towards your ankles and feet. It's quite spectacular, and also painful at the same time. So really, here we wanted to come up with a technology where we can a device that is insect repellent. Because if you use a topical repellent, for example, and apply it on your skin or apply it on the ankles, you have to reapply after 2 or 3 hours. And that's very expensive, particularly for people who live in these malaria endemic areas. It's usually resource-limited areas. So we wanted to come up with a cost-effective technique that we could make and deploy in those areas. So I don't want to dwell too much on the technical bits. I just wanted to show you that the basic idea was to produce a fiber that is specially engineered to hold the insect repellent inside. It acts as a reservoir, and it holds the liquid inside the fiber structure and slowly releases small bits of the liquid over a long period of time. Okay, so what has been released is the insect repellent that will work as a repellent and then evaporate eventually into the atmosphere. But a huge amount is still contained in the matrix for release. In this way, instead of an insect repellent formulation lasting for a few hours, you can extend it to a couple of months. An example of a slow release formulation is, for example, if you buy a very expensive cologne, such as diesel, and you spray it, you'll smell good for the whole day. But if you buy a very cheap cologne, you smell good for an hour or two, and the fragrance just quickly dies away. So that expensive cologne, it contains a formulation that slowly releases small amounts of the active so that it lasts the whole day. So this is the same principle here, but in this case, we're using textile quality fibers, and these fibers that have been infused with insect repellent, then it can be made into a textile, and we can make textile devices. So that's the fiber that has been rolled up on the left image. On the right image, if you take a strand of the fiber, you cut it across, and you look at it from the top using a very powerful microscope, the scanning electron microscope. That's what you see. You see the similar type of structure that I showed you here, a core sheath structure. And you can see on image B, that's the fiber containing the liquid inside. So it forms, in the center, it forms some small pores, small micro reservoirs. That is where the liquid is stored. And the liquid wants to come out of the polymer, but it's restricted by the polymer that wraps around the fiber. So in that way, the insect repellent is being released slowly. If you wash the textile made of that fiber, you only wash what's been released. You can't wash what's stored inside the fiber. So the textile continues to be insect repellent even after multiple washes. Okay, so this is a typical foot in cage test. Fortunately, I can't play the video. Where I wanted to show you where basically when you're testing the product, we made the fiber into a sock, and I wore the sock on one foot, and I wore an untreated sock on the other foot, and you insert your feet in a cage about 1 meter in length, half a meter in height, and half a meter in width. And in there you have 300 hungry female mosquitoes that want to feed, and they immediately go for your ankles. But the video was going to show that the females or the mosquitoes went straight for the untreated sock, and they were biting through the untreated sock, and they avoided the treated sock. And in this way, we could show that you could wash the textile for up to 25 or for at least 25 washes, and you got 100% effectiveness. And you could leave the textile in the open, or the sock in the open for up to 8 months and still exhibited acceptable residual repellent effectiveness. So upon the encouragement of Walter, I decided to start up a small biotech company, and this company manufactures insect repellent outdoor socks for people who are going into these malaria endemic areas. It is advisable to at least have your sock or your ankles if you don't like wearing socks in summer. All these products are made of the fiber that is infused with the insect repellent, and it's being slowly released by that fiber in those articles. If you are interested in more information about these products, you can send me an email. You can find them on our online shop. We're stocked out because of these COVID disruptions, but we're busy trying to get production back so that we have enough stock for the holiday season. But yes, these products are already on the market. Okay. To conclude, I'd like to thank these organizations that have supported us from the idea phase up to the current commercial phase. Thank you very much.
Thank you, Mtoko. This was really exciting. I think this is taking innovation to the next level, and also a good example of how to take research to the marketplace. We are really proud of one of our alumni addressing this issue of malaria and coming up with new innovative ideas. So thank you very much for that. Ladies and gentlemen, let's try and look at some of your questions. And the first one that I've got here is addressed to Professor Leo Brock. Leo, if you can hear me, this question is specifically dealing with climate change. How can climate change affect the species of Anopheles that is found in an area? Leo?
Thank you, Tian. I'm not sure. Can you see me and hear me?
We can hear you.
Okay, that's okay.
There you are. Please continue.
Okay. Yes, there's a genuine concern, not just for malaria vectors, but also for the Aedes species that are responsible for transmission of dengue, Zika, chikungunya, yellow fever, West Nile virus, all kinds of different arboviruses. But in the case of Anopheles, there are also already published results showing that the higher areas of certain mountains that were previously devoid of malaria vectors, villages along the slopes of mountains are now beginning to experience malaria transmission because the average temperatures are escalating so that malaria vectors are now able to survive and breed and transmit malaria at higher altitudes because of climate change-induced changes. There's also a geographic spread as global warming effects start being felt. And as I intimated, it's not just malaria. Of concern also is the impacts or the beneficial effects on especially Aedes aegypti and Aedes albopictus in transmitting these arboviruses. So I'm not going to dwell on that because this is a malaria presentation, but just think of this. There's almost 400 million cases of just dengue each year. That's a whole bunch more. That's many millions more than malaria. And although they don't cause the deaths that malaria does, it's an extremely debilitating disease of serious public health consequence. Africa, by and large, has escaped the full impact of dengue and other arboviruses, except for yellow fever. But it's a big issue in Asia, South America. You've heard about Zika in South America. It's coming to Africa. It's going to happen to Africa. It's already on the increase. So climate change does have significant, will make a significant difference on public health via mosquito-borne diseases.
Thank you, Leo. Thank you. Let's address the next question to Sarah in Montreal. Sarah, obviously, people are exposed to complex mixtures and not only a specific insecticide. So I think this question is basically dealing with that issue, asking, was simultaneous alcohol exposure excluded? Sarah?
No, it wasn't. So this is a question, I guess, asking about our populations we studied from Limpopo. It was not excluded. So again, men living anywhere will have a multitude of exposures. And, we expect there to be influences of all of them, but you try to study as realistically as you can. The animal models allow you to get at, the fine details and the mechanism because you have only your chosen exposure, and you use inbred mice models. In men, it's very complex, and you just can't do that.
Thank you very much. Indeed, a complex issue and a difficult study. The next question is to Dr. Sibanda. How can this evidence-based research on malaria contribute to government's interventions in early childhood education, since child mortality needs to be reduced? A mouthful.
I think the intervention can have a big effect in reducing child mortality. I think the most important thing is for us to show that that is the case. And one of the ways in which we're doing that is we are in the process of carrying out a field study of these products to show that they have an effect in the effect that they are supposed to have in the field. And once you have such evidence, it's very easy to approach the government or donor funders to say, hey, here is an intervention, and we think with this intervention we can reduce child mortality.
Thank you very much. I think that also stressed the importance of health promotion and education on all aspects related to malaria control and getting us eventually to malaria elimination. The next question to Leo, Professor Brock, has there been any modeling to predict the amount of decrease there will be in human vectors when Ivamectin is delivered at cattle at secondary targets?
Not as far as I am aware. So that reflects the relatively recent nature of the application of this particular new approach in the use of ivermectin. So there have been a few, and I'm talking 3, 4, 5 studies in Africa, mainly in East Africa, on either stabled cattle or in semi-field cattle in enclosures using ivermectin. then in Asia, there are currently at least 2 studies, field-based, looking at real-life situations in the application of cattle-administered Ivamectin. So, you know, there's not that wealth of data available that can be used for modeling studies, but it's being generated at present. So that is an open field for somebody to get involved in, to do those kinds of studies to predict the likely impact at scale of the application of this technique.
Thank you, Leo. I think we've got time for one last question, and this is addressed to Professor Kimmins. Sarah, have you evaluated changes in the associated chromatin proteome?
No, we haven't. So if you were to look at the proteome in sperm, you would have to go back to earlier stages in spermatogenesis to see how it affected gene expression during sperm development. And obviously we can't do that with this group of men because it would require a testes biopsy. What we have done is we've used animal models to track the sperm changes on the chromatin to the embryo. So we've looked at the chromatin in early stages of embryo development at the 8-cell stage. And what we found is that the changes that are induced by an environmental exposure are retained on that chromatin in the embryo and associated with altered gene expression during development, and then with poor reproductive outcomes, things like birth defects and metabolic disorders. So we're quite confident, based on our analysis of the chromatin in sperm in men and in embryonic gene expression in human embryos, which we've looked at data from single cell studies. There's a very strong correlation between where you have the chromatin and the changes in sperm from men and gene expression in the embryo. So we postulate that similar mechanisms are at play in men.
Thank you, Sarah. Ladies and gentlemen, I think I need to thank our speakers. I think they showcased a little bit of the University of Pretoria Institute for Sustainable Malaria Control, the diversity, the transdisciplinary approach, the interfaculty approach to a specific problem, and the commitment towards getting towards malaria elimination, supporting government, supporting the provinces, collaborating with partners towards solving these challenges that we are experiencing on a regular basis. So from Thailand, Professor Leo Braak, thank you very much. I know it's evening there. You may have a glass of wine and go to bed. Professor Sarah Kimmins from Montreal in Canada. You're allowed to have your breakfast now. Thanks for joining us and for your collaboration on these studies as well. And then also with me in the Future Africa studio, Dr. Sibanda. He is part of the UP Institute for Sustainable Malaria Control. He's been involved in the Postgraduate Student Forum. And by saying that, I need to especially thank our students for this initiative, for having these discussions, for bringing different departments, different faculties, different topics related to malaria to the table. And together we will make an impact. So thank you to you as viewers. Thank you for your contribution and your support. And I trust that you will have a good weekend. From my side, goodbye.